So we are recording. So a couple things with this. I really went through this and cut out any fluff that was in there. So really, every slide is important in this one, except for there's a series of reactions where it kind of shows all the chemical iterations, right, the process. Those you don't need to know that, but I show it to you just so you get a big picture. But this is really kind of tailored to like, this is it. Okay, so cellular respiration. When we talk about cellular respiration, we always compare it to photosynthesis, right? Cellular respiration is an exergonic or endergonic reaction. Exergonic. Is it a catabolic or anabolic process? Catabolic, breaking down, right? But the opposite of cellular respiration, you know, not quite opposite, but kind of opposite in cellular respiration is photosynthesis, right? Which is an endergonic reaction, which is an anabolic process. So photosynthesis uses light energy in order to form organic molecules, carbohydrates, sugars, G3P. How many people have heard of G3P? Glyceraldehyde 3-phosphate. You have. So that's the molecule. It's just a sugar molecule. It's a precursor to glucose. So I know we talk photosynthesis, it makes glucose. Technically, it doesn't make glucose directly. It makes a G3P molecule, which is a 3-carbon sugar. But if I put 2 3-GPs together, guess what I get? Glucose, right? So that's kind of where we are. So just realize that they're opposite processes. The reactants of one are the products of the other. And you know, these aren't new concepts. Right? You should have had this before at some point. When we talk about cellular respiration, we talk about aerobic and anaerobic respiration. Which one is more efficient? Aerobic is far more efficient, right? So cellular respiration in an aerobic environment is far more efficient. I'm just showing Homer because you can see he's undergoing cellular respiration because he's running. So therefore, the breakdown of glucose is going faster and faster to produce energy. Hence, in order to do that, we need oxygen. You're going to start breathing faster. Your heart rate goes up. Why? Because we need to get oxygen to your mitochondria for the process, right? Linking the big picture. I put a picture of plants here because I do not want my students to forget that plants undergo respiration as well. They have mitochondria. So don't forget that. On the right-hand side, we have fermentation. We actually have the ability to both. We prefer aerobic respiration. However, when you get extreme muscle fatigue, when your muscles are working too hard, you cannot keep up with the oxygen, your muscles don't die right away. You don't just go and die, right? What happens is it kicks into the process of anaerobic respiration or fermentation, which is lactic acid fermentation. Hence, he's got a lot of lactic acid going on in there because his muscles are starting to burn out. We also have alcoholic fermentation, which is done by some organisms which prefer to be anaerobic, or some that actually can go both ways as well. So when we talk about this, we're really going to talk about aerobic respiration, anaerobic respiration. Easy enough? 90% of it is aerobic. That's what we're going to talk about. The anaerobic is just a couple key points. Okay. What's the point of respiration? To make ATP. That's it. That's the point. We are taking the energy out of the very energetic glucose molecule, which is like a $10,000 bill. We're going to the bank, we're going to the mitochondria, and we're breaking it down into smaller bills, 5s, 10s, 20s, which is ATP. There's 2 ways in which we make ATP. The main way in aerobic respiration is oxidative phosphorylation. Everyone remember, way back to biology class, electron transport chain, ATP synthase, right? All of that is the oxidative phosphorylation part, which I'll talk about the details of that later. However, ATP is also made through substrate level phosphorylation. This is very, very simple. Hazel, I need a volunteer. Thank you for volunteering. Go ahead and stand up. You are ADP, okay? Right? I am a molecule with an extra phosphate group. I'm going to give you this phosphate group. You are now not ADP, you are now ATP. That's what substrate level phosphorylation is. It's a direct transfer of a phosphate group. Thank you very much. It's a direct transfer from one molecule to another, typically a glucose-based molecule or a molecule that's been broken down by glucose, and a direct transfer of a phosphate group, right? So that's substrate level phosphorylation. It's also implying that what's probably involved here. If I use the word substrate, it means what's controlled, and enzymes controlling this reaction. Okay? So that's when we make some ATP like that. Not much, but a little bit. Most of our ATP comes from the oxidative phosphorylation. This involves redox reactions. Chemistry people, redox reactions, right? Leo Gurr? All right, so we'll talk a little, we don't need to know much about redox, but we'll talk about it a little bit. All right, but these are two key terms to know. Okay, redox reactions, right, involves oxidation reduction reactions. How many people have had oxidation reduction reactions? A couple people. All right, 3 people. All right, so oxidation reduction reactions is simply the transfer of electrons. It's an energetic reaction that it's moving electrons. Really, really simple. We do not need to get too complex with this. We just need to have a basic understanding. Leo, losing electrons, oxidation. Gurr, gaining electrons, reduction. So there's how you remember it. Here's how I read it. I don't really like the Leo Gurr. I remember it like this. Right? When I gain electrons, what happens to my charge? It goes down, right? Because electrons are negative. If I gain negative electrons, my charge goes down. So therefore, I am being reduced. My charge is going down. So that's what I think about. I think about reduction as gaining electrons is going down. Therefore, the counterpart is oxidation. If you have reduction, you have oxidation. You do not have one without the other. What's that? Okay. So if you have oxidation, you must have reduction and vice versa. So reduction is when our charge is going down. Right? Oxidation is when your charge is technically going up. You're giving off electrons. You're losing electrons. Here's the thing, though. The oxidizing agent is the one that is doing the reducing. The reduction agent, the reducing agent, is the one that's doing the oxidation. A little weird, but let me explain how you remember this. Which atom, we mentioned it before, has one of the greatest affinities for electrons? Oxygen. Very good. What do you think is one of the greatest oxidizing agents out there? Oxygen, right? Oxygen is a great at reducing things. Oxygen wants to pick up electrons. Hence, oxygen is so important in this process because what picks up, like, all these electrons are being transferred. Guess what's picking up all these electrons at the end? Oxygen, okay? So that's how I remember it. Reduction is gaining electrons and charge is going down. Right? The oxidizing agent is the one that's being reduced. Remember it because oxygen is one of the atoms that wants these electrons more than almost any other one. Okay. Just kind of looking at here, it used to kind of pop up in stages. But here I have NaCl. Right? Na is losing electrons. Chlorine is gaining electrons. So chlorine is being reduced. Right? Sodium is being oxidized. Right. Here's my molecule X. Here's my molecule X. Here's my molecule Y. Here's the electron. X is losing the electron. Y is gaining the electron. So Y is being reduced. Right? X is being oxidized. Yes.
What's happened in the arrow that makes it so that happens? Why does that need to happen? You know what I mean? Like, what's the point of it? Why can't X just keep its electron?
Because there's a chemical reaction occurring. So the environment and these two chemicals, in order to get to a more stable state, are going to transfer. So think about, if I have, oxygen and water combining to form, oxygen and hydrogen combining to form water. In order to get to that stable state, electrons are going to be transferred. So that's what it is. It's getting to a more stable energy state. is what drives it. Okay, now this one's a little harder to see, but this one you just need to know, right? Here is cellular respiration. Glucose is being broken down in cellular respiration. That's what we're doing. We're breaking the energy out of glucose to make it to take that energy to put into ATP. So glucose is being oxidized. Makes a lot of sense, right? We're not going to put more electrons, more bonds onto glucose. We are going to break glucose off. So just know in this process, glucose is oxidized. Okay? And what's being reduced, or what's the oxidizing agent? It is going to end up with oxygen in the big picture. Everyone okay? Not too bad so far? All right. Have we been nuked yet, Austin? Are we okay? Okay. So oxidation, and this is kind of just a big picture one. So this is just a concept one. There's nothing to really memorize off this. We could burn glucose and just release all the energy from glucose all at once. If we do that, though, we wouldn't be able to harness all of glucose energy. A lot of that energy would be lost to heat. So what we do in cellular respiration is we don't break glucose down in one step. We break glucose down step after step after step after step. So what we're doing is we're slowly oxidizing glucose, which means we're slowly ripping electrons off of glucose, because the electrons are the bonds, the electrons are the energy. So we do it in a step-by-step process, which makes this process more efficient. If I were to do it all in one, I would get some energy, but a lot of it would just be lost to heat. By doing this step by step by step, by having this complex series of chemical reactions, which you're going to see, it makes the process more efficient. Okay, this is just an introduction so when I talk about NAD, you have a little bit of idea what it is. So remember, we're oxidizing glucose, which means we're ripping electrons off of glucose. Electrons don't do well by themselves. They're highly reactive. You put an electron in a system, that electron is going to bind to something. Okay? So when we're ripping electrons off of glucose, we can't just send them off on their own to go where we want them to go. We need something to carry them. So we have what's known as electron carriers. NAD is a key electron carrier in this process of respiration. Nicotinamide adenine dinucleotide. You will never, ever have to say that word again. You'll probably never see that word again. It's always referred to as NAD. Okay? In NAD, this is the molecule here. It's this big, complex molecule. It is a coenzyme. Right? It helps other enzymes work to be more functional. But its job is simply to carry electrons. When I'm oxidizing glucose, when I'm ripping electrons off of glucose, I need to transport those electrons somewhere. This is the ride. This is the taxi. This is how they get there. So what happens is NAD+, which is just the standing state, it's in a positive state, picks up electrons. It also picks up hydrogen as well. And it is reduced to form NADH. So NAD+, its charge is going down, is reduced to form NADH. And that is our electron carrier. We're going to talk more about this. I'm just introducing. Question? No? Okay. Okay. So, now that's the background. Now we can talk about respiration. Respiration is broken into 3 parts. Glycolysis, the citric acid cycle, or the Krebs cycle, and the electron transport chain, right? Those are the 3 processes we're going to talk about. That's what the rest of this is, these 3 processes. Glycolysis, that's the starting point. Where does glycolysis occur? It does not occur in the mitochondria, which is where respiration occurs. It occurs just outside the mitochondria in the cytoplasm. So it occurs like in the cytoplasm. From there, the molecules will move into the mitochondria, into like the middle of the mitochondria, which is the matrix of the mitochondria. I'll kind of give you some points as well, where the citric acid cycle occurs. And there at the end, we have our electron transport chain. All right, so this is just a good kind of, hey, these are the key steps of this process. And it gives you kind of the key things that are going on. And here's just another image of it. So glycolysis occurs in the cytoplasm. We start with glucose. We end up with a pyruvate molecule, which is a 3-carbon molecule. That's important to know. Pyruvate is important. And in that process, we make a little bit of ATP. We do it through the process of substrate level phosphorylation, direct transfer of phosphate groups. Then that pyruvic acid enters the mitochondria. It enters the citric acid cycle. We make a little bit more ATP. All right? What's not shown here, which I'm going to show, is not only are these processes making ATP, they're also forming a whole bunch of electron carriers. The NADs, the NADHs, the FADH2s, we haven't mentioned yet. And all those electron carriers are going to go to the ETC, the electron transport chain, which is where oxidative phosphorylation occurs. So the key thing here, just to kind of keep it back in mind, the formation of ATP is important in the first 2 steps, but the bulk of the energy is going to come from the electron carriers that we're going to form. This slide, put a star in this slide. This is what you need to know for glycolysis. Glycolysis starts with glucose. There's an energy investment phase with glycolysis, right? I'm going to start the breakdown of glucose. There's an energy investment phase to kind of kickstart it, right? I break glucose down into 2 molecules. I break it apart. I get 2 3-carbon molecules. What's kind of good to know here is this actually is G3P. G3P is the product of photosynthesis. So there's some similar molecules here. So it's not bad to know the one in the middle is G3P. Not hugely important, but understanding how they relate is a good thing. Now, what we get when we finally start breaking this down is we actually produce 4 ATPs. But we talk about this only producing 2 because there's a net gain of 2. I had to invest 2. I get 4. So really this process just gives us 2 ATPs. It also gives us 2 NADHs. So NAD plus is reduced to form NADH, right? We form 2 electron carriers. And then we get our product of pyruvic acid. Pyruvic acid is also known as pyruvate. They're the same thing. So pyruvic acid, pyruvate are the same thing. This slide is important. This is what you need to know. These are all the steps of glycolysis. So I'm going to just kind of show you the steps. Not that you need to memorize anything here. You don't need to know all these different steps. But you might need to identify where an oxidation or reduction process occurs. So that's all I'm going to do is walk you through. If you ever got a question on this, you would have all these listed out for you. You'd just be kind of analyzed. So don't freak about these next slides. This is just kind of FYI. What's that? I'll show you in a second. So we start with glucose, right? We have the energy investment phase. So therefore, we create glucose 6-phosphate. We then rearrange to fructose. We get our G3P, right? And then we get our electron carriers in our ATP forms. This is kind of taking you through the process, right? Glucose, this is the energy investment phase. So I could ask you something, I could give you the entire reaction and go, hey, where is energy invested into the reaction? You can say, well, in step 1, right, glucose is phosphorylated to form glucose 6-phosphate. You would have this listed out. You would simply just have to find it. Just have a little bit of basic understanding. We then simply, it's rearranged, right, through a series of chemical reactions, right, all of these enzymes, phosphoglucoisomerase, right? So kind of going through, another ATP is added in, so we get fructose 1,6-biphosphate, right? It's been rearranged from a 6-carbon sugar to a 5-carbon sugar. Fructose is a 5-carbon sugar. Fructose 1,6-biphosphate is simply saying that phosphate groups are on the number 1 and the number 6 carbons. Again, just get a little bit of reading, but this is just FYI. I'm not saying memorize this stuff. We kind of keep going through the process. It finally gets cleaved into 2 molecules, which is going to get us to pyruvic acid. And then here, I could say, hey, in what stage do we have an oxidation reduction process occurring? You have to find, oh, here we go. NAD plus is being reduced to form NADH. So NAD plus is being reduced. G3P, glyceraldehyde 3-phosphate, must be oxidized. Something like that. Again, you have it all here. So when we get to the final process, we create ATP. So I guess where ATP is formed. So in this step here, you have ATP being formed. And then we finally get to our pyruvic acid, our 3-carbon molecule. Again, this is what you need to know, right? How many ATPs does glycolysis produce? 2. How many electron carriers does glycolysis produce? 2 what? 2 NADHs. Very good. And what's the other product of this process? Pyruvic acid. That's what you need. Okay?
So like there's 2 carriers?
Right, so we get 2 electron carriers. Those electron carriers travel to the ETC. They produce the rest of the energy.
Each electron carrier only carries 1 electron.
A pair of electrons. So it's a pair of electrons. It comes down to NAD. For every NADH you form, you actually get 3 ATP molecules. Oh, we'll talk about that later.
What exactly happened with pyruvic acid? Is that just...
Okay, so pyruvic acid is the product of glycolysis, but it is the starting point of the Krebs cycle, the citric acid cycle.
Oh, okay.
So it's the starting point to the next one. That's why we care about pyruvic acid. So we're just kind of moving through. Right, we just finished glycolysis. We have our pyruvic acid. That pyruvic acid actually passes through the membrane of the mitochondria and actually passes to the internal part of the mitochondria. Let me just see real quick. I think I moved that slide. I'm going to add just an image up here. I think I deleted it out going forward. So on an image, just a couple key structures of the mitochondria, just structural things. The Krebs cycle, remember the mitochondria has 2 membranes, the inner membrane and the outer membrane. The Krebs cycle occurs inside the inner membrane, and that's known as the matrix of the mitochondria. So you might want to jot that down. If we do this, I can actually use some of my artistic abilities here. All right, so here's our mitochondria. Kind of looks like a kidney bean shape. Right, so this is the outer membrane. This is the inner membrane. This center region is known as the matrix of the mitochondria. That's where the Krebs cycle occurs. And all of these little folds of that membrane are known as cristae. And that's where the ETC occurs. That's where your ATP synthases are located. So just kind of give you, I'll put an image into this as well, but those are the key structures. Yes?
So you said like the reason that's cold is because it wants more surface area, so that's because of the transport chain?
The electron transport chain, exactly. The more surface area we have, the more proteins on the electron transport chain, the more ATP synthases we can pack in there, right? And we increase production. So that's a key thing, is that we have increased surface area with the inner membrane of the mitochondria to increase ATP production.
Wait, is this all synthesis?
ATP, sorry. ATP synthase. Okay, so I'm just going to work through the Krebs cycle, and then I'm going to let you kind of, well, let's do this. I want to get through the Krebs cycle. I can do it 2 ways. We have time, so this class goes to 32. Do you want me to give you 10 minutes to work on some of those handouts that are coming that are going to kind of go over this material before I talk about the Krebs cycle next part? It's not a ton to it, but there's a little bit to it. Or do you want me to just go through the Krebs cycle? We'll finish up that. We'll stop after the Krebs cycle. We won't deal with ATC today. We'll do it tomorrow. And you guys can work on the worksheets at the end of class.
Yeah.
Second option.
Heat plug. Okay, so very simple. The Krebs cycle starts with pyruvic acid. Pyruvic acid passes through the membrane to the mitochondria to the matrix of the mitochondria. And we continue to break it down. So this is kind of the simplistic form of the Krebs cycle. You do not need to memorize any of this. You just need to have a general idea of some key terms. This process, there's two ways to look at this process. This is dealing with the whole thing together, and here are our numbers. For every pyruvic acid molecule, once it enters this whole Krebs cycle, we get 4 NADHs, we get 1 FADH2. FADH2 is just another electron carrier. It's just like NADH, just another one that's involved. And we get 1 ATP produced. I turned the temperature way down. Is it too cold?
Yeah.
I was trying to wake us up. All right. So this slide is really kind of giving you the big picture here. So 4 NADHs, 1 FADH2, 1 ATP. But that is per what? Per pyruvic acid. How many pyruvic acids do we get in the glycolysis? We get 2 pyruvic acids, right? 2 of them. So these numbers could double, depending on how you ask the question. If I say, hey, how many, what do we get, what are the products of the Krebs cycle for pyruvic acid? You're going to be 114. How many for glucose? It would be 2 ATPs, 2 FADH2s, and 8 NADHs. So just realize how the question is asked. If you're talking for a glucose molecule, everything doubles, because I get 2 of them. The first part here, and again, you might want to, you're definitely going to want to jot down a few terms as we go. So the first part is really not a part of, this is known as the Krebs cycle or the citric acid cycle. It's called the citric acid cycle because the first molecule in the cycle is citric acid. It's called the Krebs cycle because the gentleman that came up with it was Dr. Krebs. That's where they two come from. So, but really it's 2 separate processes going on here. This is known as so this is just pyruvic acid oxidation or acetyl-CoA formation. Right? All it's saying is before pyruvic acid's ready to go into the citric acid cycle, we got to break it down a little bit. We got to oxidize it. So really what we do is, it's kind of hard to see the 3, but we basically take that 3-carbon molecule, right? We rip some electrons off to reduce NAD plus to NADH. Right? One of the carbons left, which makes sense because we haven't talked about it yet. It's starting here. What do we breathe out? We take in oxygen to power this, but the byproduct is carbon dioxide. So here's one of the carbon dioxide molecules that's being breathed out, right? And when that happens, I no longer have a 3-carbon molecule. I have a 2-carbon molecule, which is known as an acetyl molecule. Coenzyme A is a key coenzyme, right? So this is kind of part of an enzyme process here. So coenzyme A binds to the acetyl group and incorporates it into citric acid. So citric acid, how many carbons does citric acid have? 1, 2, 3, 4, 5, 6. So really, it's not shown, but there's a 4-carbon molecule here. It picks up the 2 from acetyl coenzyme A, and it makes the 6-carbon citric acid. So what do you need to know for that? It's just know that, hey, pyruvic acid's broken down, right? And when it's broken down, we get an NAD. An NAD plus is reduced to an NADH. Kind of a good thing to know. And acetyl-CoA, coenzyme A, helps to pull the acetyl group into citric acid. Everyone okay? Once we're citric acid, it's pretty straightforward. Again, this whole process is oxidation. So we simply rip off some electrons to form NADH. A carbon is left. A carbon is ripped off to form carbon dioxide. And we go from a 6-carbon molecule to a 5-carbon molecule. We rip off another carbon dioxide. We rip off some more electrons. We form an NAD+, and we reduce an NAD+ to an NADH. Right? And we're left with a 4-carbon molecule. This process also allows the production of ATP. You don't need to memorize the numbers. You don't need to memorize the steps. Think about the process. We're simply breaking the molecule down, releasing carbon dioxide, ripping off electrons to reduce NAD+ to NADH. That's what you need out of this. Don't memorize what each step is. Okay, just the big picture. Yes?
What prompts all of this CO2 break off?
I'll show you. I'm going to show you the real form of this in a second. So now we have a 4-carbon molecule. They're rearranged. So these 4 carbons are in like a high energy form. They're rearranged to a lower energy form. So it's a different molecule, still 4 carbons. Electrons are ripped off, so FAD becomes FADH2. That's another electron carrier. And then the cycle, then another NADH is formed, and then the cycle happens again. You don't need to memorize where things have been formed. You're not going to be asked at what point is FADH or what point is NADH. You don't need to know that. All you need to know is the molecule is being oxidized, it's being broken down. Electrons are being ripped off to reduce NAD+ to NADHs, to reduce FADs to FADH2s. Carbon dioxide is released, and in the end, you simply need to know this. That's all you need. You don't need to know exactly where it happens. You just need to know what happens. The only one you need some kind of technical, you'll be able to kind of talk about a little bit is here, is what's going on in that acetyl-CoA formation. Everyone okay? All right. I'm going to stop here, but I want to highlight the electron transport chain because how it's all tied together. This process makes about 36 to 38 ATP. I'll talk about the wiggle room there later on. Don't worry about that. We'll just call 38 ATPs. How many ATPs have we produced so far? How many do we produce in glycolysis? 2. How many do we produce per glucose in this? We've 1 per pyruvic acid, which per glucose would be 2. So we've only produced 4 ATPs. How many do we need to get to? 38. Right? So we have 34 more ATPs to go. Well, we've also produced all these electron carriers. For every electron carrier it's produced, when it goes to the ETC, the electron transport chain, they produce 3 more ATPs. That's where the work, that's where the heavy lifting happens. So just to kind of show you, and these slides here kind of walk you through the process if you want to read through. It's what I just really said. Here's the entire citric acid cycle, which I'll go back to in a second. But really, this is it. Electrons are bonds, right? That's what bonds are. Electrons are energy. So all the electrons that we've been ripping off, all that energy is going to be used in the ETC to produce ATP. I'm not going to walk you through the specifics here. I'm not going to kind of talk about it here. I'll talk about it tomorrow because I feel like you had enough. But just realize all those energy, all those electron carriers, all that energy is going to be used to make ATP here in the cristae of the mitochondria. Now, Gabby, you were asking about what's controlling all these. Well, there are enzymes at each stage. This is really what the full citric acid cycle looks like. You have your citric acid, right? You have your NADH formation, the NADH formation, ATP is being formed, and so forth. One thing I'm going to throw it out there right now, just because it's on the screen, is ATP is an adenosine triphosphate, right? It's a nucleotide. What are other nucleotides? You have A, T, C's, and G's, right? So ATP is the main one, but that guanine, see right here, it says GTP. Just know that's another energy molecule that's out there. And I'll reference it a few more times, but just be aware, just like ATP is, we we actually have a GTP molecule as well. It's an energy molecule. ATP is by far the most prevalent. Why GTP exists, I'm not quite sure. Maybe it's a relic. It's just because it's on there, that's what it is. It's just like ATP, except it's a guanine instead of an adenine on top. All right.